Right. So, hello everyone. Good morning. This is Arsene again. So today, this week, we are going to talk about the advanced tracking part in Imaris. So I'm Arsene from Taiwan. I'm sales and application specialist for Imaris, cover Australia, rest of Asia means APAC, except China and Japan. If you have any questions about Imaris, you can email me directly at ed.chang@bplan.com. Right, so here is today's learning objectives. So last week we talked about the basic tracking. We mentioned about the different algorithms and how to edit your track, how to visualize your track result. Today we will be more focused on the Lineage algorithm, which can analyze the cell division kind of applications, images. Then we are going to mention about how to use the shortest distance measurements to track the contact event between the objects. And the last one is how to use reference frame to correct the drift in the tracking, in the time-lapse images, mentioned about the drift correction. Right, so in the Lineage algorithm, there are some specific statistics you can measure, including the generations of the cells and the time-related measurements, like time since first division, time since previous division, etc., and the cell cycle duration. All these statistics related to the division event you can get from using the Lineage algorithm. So I will show you how to use Lineage on the cell division or similar image samples. Okay, so here, let me start Imaris. For the cell division kind of image, I have this example. Okay, so this is a 3D time-lapse image, and when I play the video time-lapse, you can see there are some cell division events in my image. And first thing you will notice is this rendering. So we are now in the blend mode, so you will see this opaque visualization. For different purpose, for analysis, we can try to change it back to the MIP mode that you can see more regular display. Okay, so for this one, we can try to use Lineage algorithm to track this cell division. First thing we need to do is we need to make either a spot or surface to identify the object. For this one, I'm going to use the spots. Okay, so like we have introduced in the previous week, this step is pretty much the same. So we can pick the channels and give the XY diameter. I need to measure here. So for the tracking, or for the division samples, maybe the one right after the division will be smaller, and prior to the division maybe is larger. So you still like a spot creation, you may want to use a like medium-sized cell as a reference. So here 3.39, here is 5.62. So I may use a number in between. So for example, 4 microns, and try to see if the spot detection can be successfully applied to each time point, like this. So we will have to go through the different time points. So here we can see the missing detection, which means maybe my number is too big. Here I try to reduce it. These two are quite small, so I need to check. Here, there are really small ones. So these ones are less than 3 microns. So maybe I have to put some numbers smaller to be able to detect these small cells. Okay. So if I use 3, and it still looks quite okay. So just make sure the size of the spot can cover the small cells. It's small enough to cover the small cells, but not to make a duplication in the larger cells. So 3 looks like a good number. So I guess the smallest cell in this image is about 2 microns, and the large one is about 5 microns. So I'm using like 3 to make the detection for these cells. Okay, so looks good. Looks good. So now I can go to the next step. Again, we can edit the spot for each time point manually in this step. If you find some missing spot or additional spot you want to delete, you can do the manual editing in this step by changing the mouse to the select mode and hold the shift left click to remove or add the spot. Right, go to next step. So this step, we are going to choose, very important, the correct algorithm to analyze the cell division, which is the lineage algorithm here. And we still need to enter the maximum distance and the gap size. So for the maximum distance, sometimes the maximum distance happens between, for the cell division case, happens between the division time frame. So like here, so this one, then the, okay, so it's this one. This is the middle of the parent cell, and the division happens here. So it's about 2.63. So we can still try to check other time point to see. So here, so it's roughly there. So I will put maybe 3 to start from 3. And maximum gap size, I will keep it at 2 and go to next step. All right, and we have also the filter by track duration. So we can remove a very short track. And down here, you will be able to see how many tracks you are going to keep by using this filter, keep or remove by using these filters. So for this one, I'm going to keep all and try to see the result and do some manual editing. So now the tracking has finished. Then first thing we have to do, we can turn off the 4 tracks display and change to dragon tail for a better visualization. And maybe I want to change the spot to a different color so it's a bit better visualization. And maybe try to check. Okay, so here I have 2 cells and we have some division events. So let's see if Imaris can detect this division in the lineage. So if we go to the edit tracks, then we can see this kind of tree if you use the lineage algorithm. So from the parent cell and the division events, so we can see there is a division for this one. Okay, so obviously you can see this one and has been divided into 2, and then these 2 has been divided, one of them has been divided again here. And this one, we can check if this division is like one of them is missing or is it connected to another one. So this one, it might be a false detection because it looks to me that maybe these 2 are actually, this one and this one are actually the same, from the same cell. So in this case, we need to correct it in the
split can be done indirectly in the batch process if you make a deconvolution template with the decon algorithm.
Hi, Arnold. Yep. So you mean, can it be combined with the deconvolution and this lineage track? Yes. In the batch processing, maybe we will mention about it in the future sessions, but we have a function is called batch, which can run the algorithm for multiple images at the same time. And since 9.3, we have this new batch pipeline. So you can combine the image processing together with, for example, tracking in this case. So you can do deconvolution first, then run the lineage algorithm. Then it can all be combined together into one batch pipeline, and then you can run that batch to multiple images in Imaris. So it's possible, it's possible. Okay, so here, let me try to see. So we have this track, which is from this. So now I'm going to show you how to edit the result. So here, let's say these 2, I'm not sure if you can see clear, but here we have two lines here. So one is divided into 2 like this one, and then this one, I want to link this cell to this one. This one is the ones I select here. So first, I need to change the selection from tracks to, I guess it's object. Then from here, I can multi-select this object. And let me check if it's correctly select. Let me try again. And this track is this one. and this one is this one. Right, so then let me go down. Okay, so I guess I need to select this one first. Then I need to break it. Then maybe from this one and this one. Okay, this one I need to break also. So from this one, I need to connect to and this one and this one. Sorry, I'm just trying to get this down. So I can connect this one and this one. Okay. So by selecting the multiple object, you will be able to disconnect and reconnect the. Okay, so I guess I'm missing one here. So let me try to re-edit this one again. So this one and this one, delete, and connect back this one. So this is a full track and this is another track. So I need to reconnect from this one to this one. Okay, so now I make it. So first we have to change from the tracks selection to the object. Then we can select the single object in the time point. And from where you want to edit, you just disconnect the original ones, then reconnect it to the correct one by select the object, like yellow highlighted. Then you can use in Mac is Command and in Windows is Control. So you can select the object and disconnect and reconnect. So you will be able to link them into the single division tree like this. Okay, then we can go on and check the other cells and make the edit like this using the track editor here. Right, just an example of this. Then in the statistic result, you will be able to see the specific statistic just for the lineage, including the generations. So you will be able to see the generation of the cell, as well as using the statistic code to label them. For example, label them generation. So generation here. Okay, so from 0 generation to fourth generation, you can see how many of these generation cells color coded. So it's quite easy to distinguish between the different generations, as well as using also other statistic I have here, time since division and the cell cycle duration, etc. So that is how we can use Lineage. The result we can use Lineage to get from Imaris like this. Okay, and we have this, like I mentioned in the previous week, if this statistic is changed along with the time, then we will have this graph next to the statistic, Then if you click this arrow to expand the graph here, you will have the preview of this change along with time. Then if you want to export this result, you can use this button here, export data for plotting. So you will have one column for time, another column for this statistic. You can easily make the plot in Excel, for example. Okay, so that is the lineage tracking algorithm I want to show you for the first topic.